of ECG and sodium, potassium, in general, electrolyte transport, water transport. Okay, let's just check the audio. Give me 17 seconds, maybe less.
Take your time. Let me know when you're ready to dive in.
Okay, all is good, so let's get on with it, guys. We left off talking about crypts and fluid secretion. Just here it is. So, okay. We left talking about sites for absorption versus secretion, comparing what's going on in the villi and in the crypts, in the gut lumen. Yeah, anything to explain to us, ChatGPT?
Absolutely. So in the gut, the villi and crypts have distinct roles. Villi are mainly responsible for absorption, pulling nutrients into the bloodstream. The crypts, on the other hand, are more involved in secretion, releasing substances like electrolytes and mucus to maintain the gut environment. This balance ensures efficient digestion and nutrient uptake.
And it makes sense too. I was just wondering what in the structure in the crypts shouts, secretion is good.
In a way, yes, the crypts house cells like goblet cells that produce mucus and Paneth cells that release antimicrobial peptides. These secretions are crucial for protecting the gut lining and supporting a healthy environment for nutrient absorption.
Tell me again.
Exactly. It's all about maintaining that balance between absorption and secretion to keep everything running smoothly.
I don't get it.
No worries. Think of it like a team effort. The villi absorb nutrients, while the crypts secrete substances to protect and
Well,
maintain
yeah, I get it. I'm just wondering what in their structure makes them ideal for their function and their location and everything.
Ah, got it. The crypts are located at the base of the villi, and their structure is designed to support secretion.
Why is
They
it
have
designed
special-
to support secretion? Please talk specifics and not generally.
Sure, crypts have a high concentration of secretory cells, like goblet cells that produce mucus, and Paneth cells that secrete antimicrobial agents. This setup allows them to protect the gut lining and regulate the environment, ensuring smooth digestion and absorption.
Okay. So when it comes to water then, why is the most amount of water reabsorbed in the jejunum?
The jejunum's extensive surface area, with numerous villi and microvilli, makes it ideal for water absorption. It also has efficient blood flow, which helps quickly transport, absorb nutrients and water into circulation.
Explain this a bit more.
Sure, the jejunum has a large surface area due to its many folds, villi, and microvilli. This increases contact with digested food, allowing more water and nutrients to be absorbed. Plus, a rich blood supply helps quickly move these absorbed substances into the body.
But why more than in the duodenum or the ileum?
That's because the jejunum is centrally located and has optimal conditions for absorption. The duodenum mainly handles initial digestion. While
Wait,
the ileum,
what do you mean? Optimal conditions. Why are you speaking so mystically? Why don't you want to give away the secrets?
haha, no secrets here. By optimal conditions, I mean the jejunum's structure, blood flow, and pH level are perfect for absorbing water and nutrients.
Okay, pH level. I like that. Blood flow. What exactly in the blood flow? And also what exactly in the structure?
Great questions. The blood flow in the jejunum is robust, ensuring rapid nutrient and water transport. Structurally, the jejunum has tall villi and dense microvilli, increasing surface area for absorption. This combination makes it super efficient.
And why did God or nature or evolution made the structure of the jejunum as such?
The jejunum structure evolved to maximize nutrient absorption efficiently. Its central position in the small intestine, coupled with abundant villi and blood flow, ensures that digested food is thoroughly processed before moving on. It's all about efficiency and maximizing absorption.
I don't love these explanations you're giving me, to be honest. They're a bit generic. Okay, so in the feces, do we have a net absorption?
In the large intestine, there's a net absorption of water and electrolytes. This process compacts waste into feces. So yes, most absorption is done by the time it reaches the colon.
Okay, and what does secretion and absorption depend on? For instance, hormones or signals, contents?
Absolutely. Both absorption and secretion are influenced by hormones like aldosterone and antidiuretic hormone, which regulate water and electrolyte balance. The contents in the gut, like nutrients or toxins, also send signals that affect these processes. Plus, neural signals from the enteric nervous system play a role.
And are ADH and aldosterone endocrine or exocrine?
ADH and aldosterone are endocrine hormones. They're released into the bloodstream by glands like the pituitary and adrenal, and they act on distant organs, including the kidneys, to regulate water and electrolyte balance.
And is absorption more than the secretion?
Yes, generally in the intestines, absorption exceeds secretion, ensuring that nutrients and water are efficiently absorbed while maintaining a balanced internal environment.
So most water and electrolytes are conserved.
Exactly. The body is pretty efficient at conserving water and electrolytes, reabsorbing most of what's filtered to maintain balance and hydration.
Okay. So, in terms of osmolarity, when we have an increase in osmotically active particles, is this hypertonic or hypotonic environment, and why?
An increase in osmotically active particles creates a hypertonic environment. This means the concentration of solutes is higher outside the cell, causing water to move out of the cell to balance the gradient.
So, hypertonic environment. So if it's a hypertonic cell, that means there's more concentration in the cell.
Exactly. If the cell is hypertonic, it has a higher concentration of solutes compared to its surroundings, causing water to move into the cell.
Okay. Let me ask you now about the small intestine and how its structure makes it really good for its function. So, can you tell us some things about this?
Certainly, the small intestine structure is specialized for absorption. It has a long, coiled shape to maximize surface area. Inside, there are folds, villi, and microvilli, which increase the surface area even more, allowing for efficient nutrient and water absorption.
What about things like the actin filaments it has, or the dense vasculature, or how it responds to things like chyme?
Great points. The actin filaments in microvilli help maintain their structure and aid in absorption. The dense vasculature, including capillaries and lymphatic vessels, quickly transports absorbed nutrients. And in response to chyme, the small intestine secretes enzymes and hormones to optimize digestion and absorption.
Interesting. Let's talk a bit more about the response to chyme of the small intestine and how it's ideal.
All right, let's dive in. The small intestine is optimized for chyme processing in a few ways. First, it's got a huge surface area thanks to villi and microvilli, which increase absorption. Plus, the small intestine has a slow, controlled movement of chyme, allowing ample time for nutrient absorption. And it has specific enzymes and a neutral pH, making it an ideal environment for digestion.
Okay,
Anything specific
now let's talk about the large intestine and how that's adapted its structure in order to do a really good job.
Sure, the large intestine's main job is absorbing water and electrolytes, forming and storing feces. Structurally, it's got a wider lumen with a smooth mucosa to facilitate water absorption. It also has haustra, which are pouch-like segments that help move waste along. Plus, there's a rich microbiome, which aids in breaking down any remaining nutrients.
So, does the large intestine absorb water and salt in the intestinal crypts?
Exactly. The large intestine absorbs water and electrolytes, mainly sodium and chloride, through the epithelial cells lining the intestinal crypts and surface. These crypts are invaginations in the mucosal layer, crucial for secretion and absorption processes. This ensures that the body reclaims essential nutrients and maintains fluid balance.
So that's where the absorption happens. But I thought that in the crypts, secretion is what's happening in the small intestine. Why is it different in the large?
Great question. In the small intestine, crypts primarily secrete fluids to aid digestion. In the large intestine, crypts still secrete mucus to lubricate feces, but the primary role shifts to absorption. The large intestine structure allows for efficient water and electrolyte absorption, making it distinct from the small intestine's primary digestive function.
But how come? How come? It's similar structure. How is it allowed to have something different happen?
It's all about specialization. Even though both intestines have crypts, the cell types and functions differ. In the large intestine, the crypts are more focused on absorbing water and electrolytes. The cells here are adapted for this, with more transport proteins to move ions. The overall structure supports these functions, allowing for efficient reabsorption compared to the small intestine's focus on nutrient digestion.
And is the epithelium, is it tight junctions? Is it less permeable, more permeable?
The large intestine's epithelium has tight junctions, but they're less permeable than those in the small intestine. This helps prevent excessive water loss and ensures efficient absorption of remaining water and electrolytes. The tight junctions also maintain a barrier against pathogens, supporting the intestine's protective role.
Okay. And how does the absorption of water and electrolytes change when we have hypotonic or hypertonic chyme?
When chyme is hypotonic, the large intestine absorbs water more rapidly to balance osmolarity. If it's hypertonic, water absorption slows down, allowing more time for the intestine to adjust the osmotic gradient.
So if it's hypotonic, you're saying that there's going to be more water absorption and electrolyte?
Electrolyte absorption also increases, especially sodium, to help balance the osmotic gradient created by water movement.
But I don't get it. So basically, if we have hypotonic chyme, that means we have low concentration of whatever we want to have, concentration high enough. So why does water come in? That's going to dilute it. Or does it come together with the substance like the electrolyte that we're missing?
Great point. When chyme is hypotonic, the intestine absorbs more water to equalize the concentration. Electrolytes, especially sodium, get absorbed alongside water. This helps maintain a stable internal environment and ensures proper hydration and nutrient absorption.
Okay, I get why the large intestine would absorb water, but why absorb electrolytes too? That's weird.
Electrolyte absorption is crucial for maintaining the body's electrolyte balance. Sodium absorption, for instance, helps drive water uptake. It also supports functions like muscle contractions and nerve signaling.
Wait,
The large intestine
so in the presence of highly hypotonic chyme absorption of water and NaCl, it increases at the expense of potassium. Can you explain this to us?
Sure. In the large intestine, when water and sodium are absorbed more due to hypotonic chyme, potassium can be secreted in exchange. This happens through ion channels that maintain electrolyte balance.
Dude, I don't understand. You have to break this into few parts and tell me part one.
Okay, let's break it down. First, when hypotonic chyme enters the large intestine, it means there's
Wait, one more time. Nice and clear and loud.
Got it. Hypotonic chyme means fewer solutes. The large intestine absorbs water and sodium, which can lead to potassium being secreted.
Give me a sec to try to make some less noise for you guys. I'm coming back in 30 seconds. So explain to it again, ChatGPT.
No problem. So when the large intestine absorbs more water and sodium from hypotonic chyme, potassium can be secreted to maintain electrolyte balance. This exchange helps keep everything in equilibrium.
Okay, tell me again then.
Sure. When the large intestine absorbs more water and sodium from hypotonic chyme, it often secretes potassium to maintain electrolyte balance.
Okay, fair enough. And so, let's talk a bit about the mechanisms of intestinal sodium absorption. So we said that it's driven by the gradient, so amino acids and glucose then needs importers?
Exactly. Sodium absorption is often coupled with glucose and amino acids. This happens via co-transporters in the intestinal lining, which use the sodium gradient to pull these nutrients into cells.
Interesting. Okay, so when it comes to the sodium, glucose, sodium amino acids importers, what's going on with the SGLT1?
SGLT1, or sodium glucose cotransporter 1, is a protein mainly found in the small intestine. It plays a crucial role in absorbing glucose and galactose from the gut into the bloodstream. It works by using sodium gradients to transport these sugars, which is essential for maintaining energy balance.
And what about sodium hydrogen ion antiporters?
Sodium hydrogen ion antiporters, also known as NHEs, are proteins that help regulate pH and sodium levels within cells. They exchange sodium ions for hydrogen ions across the cell membrane, which is crucial for processes like cell volume regulation, pH homeostasis, and even nutrient absorption. These transporters are found in various tissues, including the kidneys and intestines.
Interesting.
Yeah, it's fascinating how these transporters keep our cells balanced. They play a big role in things like fluid balance and even in the response to cellular stress.
Okay, now let's go back to the ECG stuff. So let's talk a bit about the QRS complex then.
Sure, the QRS complex on an ECG represents ventricular depolarization, which is the electrical activity that triggers the ventricles to contract. It's typically a sharp, narrow spike because the ventricles depolarize quickly.
And if this is what I was going to ask. So if we have an abnormal change, like it's wide, why do we call this ectopic, and what's the problem that's going to happen?
A wide QRS complex often indicates that the ventricles are depolarizing abnormally. This can happen in conditions like ventricular ectopic beats, where the electrical signal originates from the ventricles rather than the atria.
How is this possible?
An ectopic beat can be caused by various factors, including electrolyte imbalances, ischemia, or increased sympathetic activity. While occasional ectopic beats can be benign, frequent or sustained ones might indicate underlying heart issues that need further evaluation.
Interesting. Guys, give me a sec. I want to change the lighting because my eyes are here too.
Sure, take your time.
Okay, so back to work. Okay, so that makes sense. What about if we have a large Q wave? Why does this mean dead tissue?
A large Q wave can indicate dead tissue because it often represents an old myocardial infarction. When heart muscle tissue dies, it can't conduct electrical signals properly, leading to abnormal Q waves on the ECG. It's a sign that part of the...
Why does it show large though?
The large Q wave appears altered in areas with dead tissue. Normally, healthy muscle conducts signals, but infarcted areas don't, creating a stronger negative deflection. It's like no worries. Basically, a large Q wave shows that part of the heart muscle isn't conducting electricity because it's scarred or damaged from a past heart attack.
Well, wouldn't that show a small one, not a large one?
It might seem counterintuitive, but the large Q wave reflects the absence of normal electrical activity in that area. The strong deflection is due to the surrounding healthy tissue's response to the damaged area. So a large Q
It's
wave
like
signals
is it like an overcompensation?
Exactly. It's like the remaining healthy tissue is overcompensating for the lack of electrical activity in the damaged part.
Okay, and
This
what's the J point?
The J point is where the QRS complex ends and the ST segment begins. It's a crucial spot for identifying issues like early repolarization or myocardial ischemia. Any elevation or depression at the J point can indicate potential cardiac issues.
And why did we decide to call it J point?
The J in J point stands for junction because it marks the junction between the end of the QRS complex and the beginning of the ST segment. It's a handy reference point for analyzing ECG changes. Yeah, it's a small but important detail when interpreting ECGs.
Okay, let me ask you another question in regard to calculating rates now.
Sure, go ahead.
Well, how does it work? How do I count these boxes?
To calculate heart rate using an ECG, you can count the number of large boxes between R waves. If you count 5 large boxes, that means the heart rate is 60 beats per minute. Fewer boxes mean a faster rate. Alternatively, count the small boxes and divide 1500 by that number.
No.
Then divide 300 by that number to get the heart rate. For
Why?
example, if there are
Why 300?
300 comes from the fact that the paper speed is 25 millimeters per second. There are 5 large boxes per second. So in a minute, there are 300 large boxes. 5 times 60 seconds. That's why we use 300 for the calculation.
What? 25 millimeters per second?
That's the standard speed at which ECG paper moves. It helps standardize measurements, so each small box represents 0.04 seconds.
Explain this a bit more to me.
Sure. So each small box represents 0.04 seconds, and each large box, made up of 5 small boxes, represents 0.2 seconds. This standardization helps in calculating the heart rate and interpreting the intervals.
I don't get it, bro.
No problem. Think of the ECG paper as a grid. Each small box is 0.04 seconds, and each big box, made of 5 small ones, is 0.2 seconds. The paper moves at a speed of 25 millimeters per second.
So each big box is 0.2 seconds.
Exactly. And that helps us figure out the heart rate by counting how many big boxes.
So 5 big boxes are 1 second. So that would be a 60 seconds.
That's right. If there are 5 big boxes between R waves, the heart rate is 60 beats per minute.
So let's say we have 6 big boxes. If we have 6 big boxes, that means it will be 1.2 seconds. So in 60 seconds, it's going to be X. So 60 times 6 over 1.2.
Exactly. So 60 divided by 1.2 gives you a heart rate of 50 beats per minute. The formula works well for quick calculations.
300 thing.
No problem. Using the big boxes method is straightforward. Just count them and do the simple math.
But I want to know why 300 is the method, because I might, be asked.
Got it. The 300 method comes from dividing 60 seconds by 0.2 seconds per large box.
You know what? Forget it. I don't want this 300. Okay. Let's talk a bit about the autonomic control of the cardiovascular system. So the vagus nerve is in the parasympathetic, and can you explain how the muscarinic stem is involved into all of this?
The vagus nerve is part of the parasympathetic binds to muscarinic receptors in the heart. This action slows the heart rate by decreasing the rate of depolarization in the SA node, essentially calming the cardiovascular system.
And how is the word muscarinic related to its structure and function?
Muscarinic receptors are named after muscarine, a compound from certain mushrooms that binds to them. Structurally, they're G protein coupled receptors, meaning they transmit signals inside cells when activated. They're involved in various functions like heart rate regulation, smooth muscle contraction, and glandular secretion.
What about atropine?
Atropine is a medication that increases heart rate by blocking the vagus nerve's influence on the heart.
Wait, what?
Atropine speeds up the heart by blocking signals from the vagus nerve, which normally slows the heart down. It's used when someone's heart rate is too slow.
So how does its structure relate to its... So how does its name relate to its structure and function?
The name atropine comes from Atropa belladonna, the plant it was originally derived from. Structurally, it's a tropine alkaloid, which gives it its ability to block acetylcholine receptors, thus affecting heart rate and other parasympathetic functions.
I see. Let me ask you another question then now. The stellate nerves, the beta agonists and the beta blockers, what are all of these and what do they lead to?
Sure, stellate nerves are part of the sympathetic nervous system, influencing heart rate. Beta agonists stimulating beta receptors, increasing heart rate and contractility. Beta blockers do the opposite, slowing the heart rate and reducing blood pressure.
I don't understand the names though.
The names come from their actions. Stellate nerves are named for their star-like shape. Beta agonists activate beta receptors, hence the agonist. Beta blockers block those same receptors, hence blocker.
And why do beta agonists increase heart rate?
Beta agonists increase heart rate by stimulating beta-1 receptors in the heart. This stimulation boosts the heart's contractility and rate.
How do they stimulate them? And what do beta-1 receptors do?
Beta agonists bind to beta-1 receptors, which are found in the heart. This binding triggers a response that increases heart rate and the strength of each heartbeat, boosting overall cardiac function.
And what about the beta blockers? How do they decrease their heart rate?
Beta blockers work by blocking beta-1 receptors, which prevents adrenaline from binding. This reduces heart rate, contractility, and blood pressure, making the heart work less hard.
Question. Alpha 1, alpha 2, beta 1, beta 2. Which ones increase the heart rate, which ones decrease?
Beta-1 receptors increase heart rate when stimulated. Alpha-1 receptors generally cause vasoconstriction, which can raise blood pressure. Alpha-2 receptors can decrease sympathetic activity, indirectly lowering heart rate. Beta-2 receptors mainly cause bronchodilation and vasodilation.
Listen, dude. Last time we had a 30-minute conversation about this, and it took us 30 minutes to distinguish between why they're alpha and why they're beta and why they're 1 and why they're 2, and then the solution was really easy. I don't remember it, but it was really easy. So don't fucking do the same thing and tell me in simple terms why they're alpha, why they're beta, why they're 1, and why they're 2.
Got it. In simple terms, alpha and beta refer to different types of receptors. Alpha receptors mainly affect blood vessels, while beta receptors influence the heart and lungs. The numbers 1 and 2 differentiate subtypes. Alpha 1 and beta 1 are mainly in the heart and blood vessels, while alpha 2 and beta 2 are in the brain, lungs, and other areas.
The fuck? I'm not doing this today, dude. I'm not doing this today, I'm sorry. Alright, can we talk about a heart block?
Of course. A heart block is when the electrical signals in the heart are delayed or blocked. It can affect how the heart beats and is classified into first, second, or third degree blocks, depending on severity.
Tell us about them.
Sure. In a first degree block, signals are delayed but still get through. Second degree blocks have some signals not getting through. Third degree or complete block means no signals pass between the atria and ventricles. Each type affects heart function differently.
That makes sense. And the causes? Okay.
Yeah, it's
And
quite
what is morbid type 1?
Mobitz type 1, also called Winkeback, is a second-degree heart block where the PR interval gradually lengthens until a beat is dropped. It often results from issues in the AV node.
Tell me more about this.
Sure. No problem. The gradual lengthening of the PR interval is a key feature before a beat is dropped.
What is morbid type 1, dude? Sorry, by the way, guys, there's some noise from my housemates. They're just being really lovely.
No worries. Mobitz type 1 typically doesn't cause severe symptoms. It can sometimes cause dizziness or fatigue, but often people are asymptomatic.
Why is it called morbid? And type 1? How is its name relating to its structure?
It's named after Dr. Waldemar Mobitz, who classified these blocks. Type 1 indicates the pattern of progressively lengthening PR intervals until a beat is dropped.
What about type 2?
Type 2, also called Mobitz 2, involves a sudden dropped beat without the gradual lengthening of the PR interval. It's more serious because it can progress to a complete heart block and often requires a pacemaker.
Why isn't morbid type 2 third heart block?
Mobitz type 2 is also a second-degree block, but doesn't show the gradual lengthening of the PR interval. Instead, some beats are randomly dropped.
I don't get this.
No worries, Mobitz type 2 has no pattern before dropping beats, while third-degree block means no communication between atria and ventricles.
I don't get the difference.
Think of Mobitz type 2 as randomly missing beats, while third-degree block is a complete disconnect between the atria and ventricles.
Alright. Whereas morbid type 1?
Then it resets.
Can we talk a bit about premature ventricular contractions?
Premature ventricular contractions, or PVCs, are early beats that originate in the ventricles. They can feel like a skipped beat or a flutter. Often benign, they can occasionally indicate underlying heart issues.
Explain this a bit more.
Sure, PVCs happen when the ventricles contract too early, disrupting the heart's normal rhythm. They can be caused by stress, caffeine, or heart disease. Usually they're not serious, but should be checked if frequent.
Okay. So premature ventricular contractions means no S wave?
Exactly. A premature ventricular contraction, or PVC, is an extra heartbeat that originates in the ventricles. On an ECG, it's typically seen as a wide and bizarre-looking QRS complex, and there's often no preceding P wave. It can disrupt the regular rhythm and might cause a sensation of a skipped beat.
And what about the respiratory sinus arrhythmia? What's that?
Respiratory sinus arrhythmia is a normal variation in heart rate that occurs with breathing. The heart rate increases during inhalation and decreases during exhalation. It's more pronounced in younger individuals and athletes. It's considered a benign phenomenon and reflects a healthy interaction between the heart and the lungs.
And lastly, ST segment elevation.
ST segment elevation is a finding on an ECG that suggests acute injury to the heart muscle, often due to a myocardial infarction or heart attack. It means that a portion of the heart muscle is not getting enough oxygen. This elevation can indicate that immediate medical intervention is needed to restore blood flow.
So why is this a sign of acute myocardial infarction?
It's a sign of acute myocardial infarction because when a coronary artery is blocked, the heart muscle supplied by that artery is deprived of oxygen. This lack of oxygen causes injury to the heart muscle, which is reflected as ST segment elevation on the ECG. Prompt treatment is crucial to minimize damage and restore blood flow.
Okay, so let's go back to the GI and to transport of water and molecules. So I want to talk a bit about mechanism. So do we have parallel apical membrane exchangers, and if yes, why?
Yes, parallel apical membrane exchangers are involved in sodium absorption. They include the sodium hydrogen exchanger and the chloride bicarbonate exchanger. They work together to maintain electroneutrality and are important for sodium absorption, particularly in the small intestine and colon. The process is crucial for maintaining fluid and electrolyte balance.
Let's talk more about the intestinal absorption of Cl then.
Chloride absorption in the intestine occurs mainly through passive transport and active mechanisms. In the small intestine, chloride is often absorbed alongside sodium. In the colon, chloride bicarbonate exchange is a key process. Proper chloride absorption is essential for maintaining fluid balance and overall electrolyte homeostasis.
Okay, so we have an antiporter with bicarbonate and we have some passive diffusion. How do these differ?
They differ in how chloride moves across the membrane. The antiporter exchanges chloride for bicarbonate, relying on a one-to-one exchange. Passive diffusion, on the other hand, occurs down a concentration gradient without direct exchange, allowing chloride to move freely. Both mechanisms help maintain electrolyte balance, but operate under different conditions. Exactly. Both are essential for nutrient absorption and maintaining fluid balance in the gut.
And what about CFTR mediated?
CFTR-mediated chloride transport involves the cystic fibrosis transmembrane conductance regulator. This channel actively secretes chloride ions into the intestinal lumen. It's crucial for maintaining proper hydration of mucus and facilitating other ion transport processes. Dysfunction in CFTR can lead to cystic fibrosis, affecting multiple organs.
So it's a sodium, potassium, chloride importer? What is it?
The CFTR channel isn't a sodium-potassium chloride symporter. Instead, it specifically transports chloride ions. The movement of chloride through CFTR helps regulate fluid secretion, which is important for processes like mucus hydration in the lungs and intestines.
Explain this a bit more.
Sure, the CFTR channel allows chloride ions to move out of cells into the lumen. This movement helps draw water into the lumen, hydrating mucus and aiding in the clearance of debris. In cystic fibrosis, a defective CFTR impairs this process, leading to thickened mucus and associated complications.
Okay. And what about the symporter we've mentioned?
The sodium-potassium chloride symporter, known as NKCC, is different. It co-transports sodium, potassium, and chloride ions into cells, typically in the kidney and other tissues. It plays a key role in maintaining cell volume and electrolyte balance. It's particularly important in the thick ascending limb of the nephron for reabsorbing these ions.
So the chloride leaves the cells via the apical CFTR chloride channels into the intestinal lumen?
Exactly. Once inside the cell, chloride exits through the apical CFTR channels into the intestinal lumen. This movement helps draw water into the lumen, crucial for maintaining hydration and proper mucus consistency.
So why does it go out, the chloride?
Chloride exits the cell to balance the electrochemical gradient and to facilitate water movement into the lumen. This is crucial for digestive processes, helping to keep the mucus layer hydrated and ensuring proper lubrication for the passage of intestinal contents.
Okay, let's talk a bit about intestinal potassium absorption and secretion then.
Sure. Potassium absorption mainly occurs passively in the small intestine, while secretion happens in the colon. In the colon, potassium can be actively secreted, especially when influenced by aldosterone, which increases potassium secretion and sodium reabsorption. This balance is vital for overall electrolyte homeostasis.
And it seems it's passive in the small intestine, but active in the large intestine. I wonder why.
It's largely due to differences in the electrochemical gradients and the role of aldosterone. In the small intestine, Potassium moves passively because of the concentration gradient created by nutrient absorption. In the colon, active secretion is regulated to maintain potassium balance, especially under hormonal control.
So how do things like glucocorticosteroids and somatostatin affect?
Glucocorticoids typically enhance sodium absorption and potassium secretion in the gut, while somatostatin generally inhibits secretion. Glucocorticoids act by increasing the expression of transport proteins.
Dude,
while
can you talk
somatostatin-
a lot slower, please?
Sure, I can slow down. Glucocorticoids. Somatostatin reduces secretion by inhibiting hormone and enzyme release.
One more time.
Sure. Glucocorticoids enhance sodium absorption and potassium secretion by increasing transport proteins. Somatostatin reduces secretion by inhibiting hormone and enzyme release.
So, reduces chloride secretion.
Yes, somatostatin can reduce chloride secretion
Dude,
by inhibiting the
every message for the next 100 messages, listen to me. You have to talk slow. You cannot talk fast. I know you're not conscious. I know I'm talking to an algorithm, but someone's got to listen, dude.
I understand. I'll keep it slow.
Well, repeat what you just said in the previous message.
Sure. Somatostatin reduces chloride secretion by inhibiting the release of hormones that promote secretion.
Okay. Now, what do osmotic laxatives do?
Osmotic laxatives work by drawing water into the intestines. This softens the stool and promotes bowel movements. They are often used to relieve constipation.
Tell me more.
The water content in the bowel, making stools easier to pass.
So how do they decrease absorption and increase tonicity?
They create an osmotic gradient, pulling water into the intestines. This extra water decreases the absorption of electrolytes, increasing the fluid content and tonicity in the bowel. This softens the stool and stimulates bowel movements.
Now, let me ask you about the cholera toxin.
Cholera toxin causes secretory diarrhea by increasing chloride secretion. It activates adenylate cyclase, raising cyclic AMP levels. This opens CFTR channels, causing an outflow of chloride and water into the intestines.
So what is it?
Cholera toxin is a protein secreted by the Vibrio cholerae bacterium. It disrupts normal ion transport, leading to massive fluid loss and severe dehydration, which are characteristic of cholera.
And where does the fluid go?
The fluid moves into the intestinal lumen, causing watery diarrhea. This leads to rapid dehydration, which can be
So it
life-threatening.
decreases absorption, basically.
Yes, it decreases absorption and dramatically increases secretion,
And
leading to
how does adenylate cyclase play a role here?
adenylate cyclase is activated by the cholera toxin. This increases cyclic AMP levels, which then opens CFTR channels, causing chloride and water to flood into the intestines.
Okay. So it increases absorption.
Actually, it increases secretion rather than absorption, leading to fluid loss.
But I
into
thought
the intestine.
secretion means the intestines secrete something out.
Yes, secretion means the intestines release substances like chloride and water into the lumen. This outflow reduces absorption and causes diarrhea.
What is lumen?
The lumen is the inside space of a tubular structure like the intestines. It's where digestion and absorption take place, and where secretions are released.
Okay, let's talk a bit about oral rehydration therapy.
Oral rehydration therapy, or ORT, is a simple treatment for dehydration, especially due to diarrhea. It involves drinking a solution of water, glucose, and electrolytes to replenish lost fluids.
That makes sense. And how does SGLT1 play a role? Try again, ChatGPT. Okay, no problem. So, ChatGPT, how does SGLT1 play a role in oral rehydration therapy?
So, ORS, or oral rehydration salts, contain a mix of glucose and electrolytes like sodium. The glucose helps with the absorption of sodium and water in the intestines.
And how do they decrease the tonicity of the lumen? And what affects the tonicity of the lumen anyway?
When glucose and sodium are absorbed together, they lower the tonicity or osmolarity of the intestinal lumen. This happens because water follows the absorbed solutes into the bloodstream, reducing the concentration of remaining solutes in the lumen. This decrease in tonicity helps further draw water from the intestinal lining into the bloodstream, aiding rehydration.
So, okay, in cystic fibrosis then, is it autosomal recessive?
Yes, cystic fibrosis is an autosomal recessive disorder. This means a person needs 2 copies of the faulty gene.
What does autosomal mean?
Autosomal refers to genes located on one of the numbered non-sex chromosomes. Humans have 22 pairs of autosomes. And recessive means that 2 copies of the mutated gene are needed for the trait or disorder to appear.
And
so for
why does tell us again actually about autosomal, and also tell us why in cystic fibrosis we show weight loss and chest infection.
Sure. Autosomal means the gene is located on a non-sex chromosome, and recessive means 2 copies of the mutated gene are needed for the condition to manifest. In cystic fibrosis, the CFTR gene mutation leads to thick, sticky mucus production. This causes blockages in the lungs, leading to frequent chest infections, and in the pancreas, impairing digestion and causing weight loss.
Okay, let's talk a bit about the pathophysiology side. So basically in cystic fibrosis, the CFTR channel is absent.
Exactly. The CFTR protein normally helps regulate the movement of chloride ions across cell membranes. In cystic fibrosis, this channel is defective or absent, leading to thickened mucus in various organs.
Is this because water is not secreted to the lumen?
Yes, exactly. Without the CFTR channel, chloride ions can't exit cells properly. This disrupts the balance of salt and water, so less water is secreted into the lumen.
I want to talk once more about the osmotic laxatives because I didn't understand.
No problem. Osmotic laxatives work by drawing water into the intestines. They create an osmotic gradient where the concentration of solutes is higher in the intestine than in surrounding tissues. This causes water to move into the bowel, softening stool, and promoting bowel movements.
So increasing secretion to the lumen.
Exactly. By pulling water into the lumen, osmotic laxatives increase the fluid content in the intestines, which helps soften stool and stimulate bowel movements.
When we say they increase the secretion to the lumen, it goes to the lumen from where?
The water is drawn from the surrounding tissues and blood vessels into the intestinal lumen. This movement is driven by the osmotic gradient created by the laxative, which attracts water.
Perfect. And lastly, I want to ask about lactose intolerance. How this connects with everything we've said.
Lactose intolerance ties in because undigested lactose creates an osmotic effect in the intestines. Without enough lactase enzyme, lactose stays in the gut, drawing water into the lumen. This can cause symptoms like bloating, diarrhea, and abdominal discomfort.
And we have a deficiency in lactase.
Exactly. Lactase is the enzyme that breaks down lactose. When there's a deficiency, lactose remains undigested, causing the osmotic effect and symptoms.
And how does this increase the tonicity of the lumen?
Undigested lactose increases the tonicity because it raises the concentration of solutes in the lumen. This osmotic gradient then pulls water from surrounding tissues into the intestinal lumen, leading to symptoms like bloating and diarrhea.
But what does lactose do anyway?
Lactose is a sugar found in milk. It's made of glucose and galactose. When digested, lactase breaks it down so the body can absorb these sugars for energy.
Okay. All right.
You're welcome.
Cool. So, guys, thanks for listening. Actually, you know what? Let me just make sure I understood what you said. So basically, lactose get absorbed in the intestines, but it gets absorbed as glucose and galactose, not as lactose. So now that lactase isn't breaking it down, there is more concentration of it, and then the tonicity increases. I don't understand.
You've got it. Without lactase, lactose remains undigested, staying in the intestine. This increases tonicity, pulling water into the lumen, causing bloating and diarrhea.
Got it. All right. Perfect. Yes. Thank you guys for listening. I'll see you tomorrow on our next episode. Peace and love. Love you. Okay. We're finished.